Okay. We're talking about DNA replication. So what we have started is known as molecular biology. Molecular biology. Now, when you're talking about molecular biology, it involves different steps. Okay. That is how exam questions start like that. So just understanding the components for molecular biology. So molecular biology, we have got what we call the central dogma of molecular biology. You need to know what it states. Central dogma. You know, a dogma is just what can I even say? More like a law, a writing, something that has been stipulated. Okay. So we're talking about the central dogma of molecular biology. What really does it say? So it all starts with DNA. Now you know that DNA is found in the nucleus, right? So DNA in the nucleus can actually make a copy of itself. Imagine you are making your sperms and you are making your eggs, the ovum. So these things will get the same DNA that is there in the mother, all right? And in the father. So what happens is that we make a copy of DNA. So we are going to convert DNA into DNA. Now remember that eukaryotic cells have got DNA inside the nucleus. So the meaning of that is that this process, DNA to DNA, for eukaryotic cells is happening in the nucleus. Prokaryotic cells don't have a nucleus, and so this step that I've written here is going to be happening in the cytosol, okay, the cytoplasm. Now, when we have got DNA, we can convert DNA now. Remember, at the end of it, or you are forming a baby, right? A baby has got hair, has got nails, has got the hands. So all those parts of the body that we're talking about are basically made up of proteins. So before you can form proteins, you first have to convert the DNA into RNA. So this is the second step of molecular biology. Now the RNA that you have formed, you can now convert the RNA into your proteins. And that is the third step of molecular biology. So you make a copy of DNA. After that, you convert DNA to RNA and then the RNA to proteins. Now, when you're making a copy of DNA to DNA, making a copy, this process is known as DNA replication. To replicate is just to make a copy. And then when you're converting DNA into RNA, this process is known as transcription. You are transcribing you are converting the DNA into RNA, so that's called transcription. Okay? Transcription. When you convert RNA into proteins, this step is known as translation. So these three steps I've written here, we call them the central dogma of molecular biology. DNA replication, transcription, translation. This is the order. So sometimes they can put an option, they put this word and then they put RNA transcript. Just know how the order should flow. So for replication is DNA and DNA, transcription DNA to RNA, translation RNA to proteins. Are we good so far? Are we good? Okay. Now we are just from discussing nucleic acids in biology. We did discuss nucleic acids and we said that DNA is a double stranded molecule. Let me not write it like this. Let me put it like so. DNA is a double stranded molecule. It's like this. And remember what we mentioned? We said that it has got its two strands, and these strands, one strand is running from 3 prime to 5 prime direction. The other one is moving in the opposite direction, 5 prime to 3 prime direction. Okay. So the first step that of the three steps that we'll discuss each one is a topic. If we discuss these three, then we are done with molecular biology. Then the next topic is genetics. Then we are done with the entire biology. We start now revising past papers for exam. Okay. So we are discussing today DNA replication. We want to make a copy of DNA. So this is the DNA that we have. We want to make a copy of it. So what should be the first thing? So the discussion we are talking about is DNA replication, not in eukaryotic cells, but in prokaryotic cells. In eukaryotic cells, it's a bit of complex, but it's similar. So we're discussing the process in prokaryotic cells. So meaning that this process is happening in the cytosol, because if it was eukaryotic cells, it would be happening in the nucleus. So this is DNA, right? So DNA replication is a process by which DNA makes a copy of itself during cell division. Okay, the cells are dividing. So let's say you started with one cell and it had DNA like this. If you want this cell to divide, the first thing that should happen is that first the nucleus has to divide, the DNA has to divide. And then after that, now you can have two different cells, each one with DNA. Okay? So that is DNA replication. Because minus this happening, that's how you end up with children who are born with genetic defects. For example, someone can be born without a mouth, someone is born without a nose. That is because there was a problem when it came to DNA replication. Some genes were, we didn't make a copy of some genes. Okay. So when you're talking about this process, again, another thing that you need to know is that before DNA replication can happen, something has to happen to the DNA itself. And what should happen to the DNA is that you need to separate it, because it is a double stranded molecule. You can see it has got two strands. So we need to separate the two strands of DNA. And what happens is that we are going to have some enzymes which are going to be able to do the work of separation, to separate the DNA strand. So when you are separating the DNA strand, you are going to have an enzyme that is going to come here. Okay, and then it will start going forward, it will start going in this direction. And so what you'll be able to see is that the molecule is going to separate. So you've seen what has happened. So here is the enzyme that we are talking about, this enzyme here. Or know the name of the enzyme. Let's say this is 3 prime, this is 5 prime, this is 5 prime, this is 3 prime. Okay, so where this enzyme, the direction in which this enzyme is going, we call it as a direction of the replication fork. You can see like this thing that I've drawn is looking like a fork, right? So we say direction of the replication fork. So which one is the direction of the replication fork? It's forward. Please don't miss this. Has anyone missed anything up to this end? Please be free to ask. I know where this thing is going. Has anyone missed anything up to here? Okay. Now, when let me go to the next page. When these biologists were suggesting how DNA replicates, so I'm going to mention the name. I'll come to the enzymes here. We have got different enzymes. So when biologists were trying to see how DNA replication happens, they proposed different ways. Okay, they proposed different ways. One way that there are three ways which were proposed. Okay, and we need to understand each one of them and which one was the true one. Is there another question before I go to this one? Nothing? Okay. Okay. So, so we are saying that the three methods were suggested of how DNA replicates. So the first suggestion which was given was that DNA will undergo what is known as a conservative replication. I'm talking about them all, but then you're supposed to understand which one is the one, okay? So one proposal which was made is that DNA undergoes conservative replication. What does conservative mean? Conservative means that for this strand that we are starting with, this chain that we are starting with, you are going to separate the two chains. You've seen? You separate the two chains like that. And then each, according to the conservative method or the conservative model, to conserve means that you are keeping, you are not really changing anything. So according to the conservative model, they said that after replication has happened, the molecule that you are going to end up forming, the DNA is going to be the same as the DNA of the, actually something like this is going to happen. Please closely look at what I'm doing. Okay. Okay, let me put it like this. You've seen, please don't forget this is the original. This is the DNA for the parent that we are starting with. So according to the conservative model, they said that when DNA is making a copy of itself, you are going to have one copy that is exactly like the original one. And then another copy which is fully new. So the two colors represent. So you've seen. So this is the child. The child is going to have this DNA. The parent has got this DNA. So I've seen that the DNA of the child is fully new. That is conservative replication. That's what they suggested. I wanted to do the conservative replication. After another replication, again, if another replication happens, this one. So every time we are always forming something that is new. Okay. Remember at the beginning we started with this one, isn't it? So meaning that these two guys, if they undergo another replication, you are going to form, this one will form one new chain, okay, like that, and then it will again form a new chain. So after every replication you are forming a new chain and you are still maintaining an odd chain, and an odd chain is like this. So if this one also undergoes replication, it is going to form, so we're going to separate the two strands, right? The two strands. So we'll first maintain the old chain. Remember the old chain is looking what? Red. But then the new chain, after every replication, the new chains are always looking red, right? At the beginning, we started with the blue one. So again, a new chain will also look red. So in other words, red is always new. So at the end of it all, the DNA of the parent will remain constant. But then you are just forming new DNA that is totally different from the DNA of the parent. That was the first model. The next model that was proposed is known as the dispensive model. Dispersive model. According to the dispersive model, what they said is that when DNA is undergoing replication, really what will happen is that some parts of the DNA is going to break. You are not really changing anything. So for example, the way this one is supposed to be like this. So some parts of it is going to break. You can see the way I'm drawing it. And then you form another chain also like that. At the end of the division, you always have two chains. So this was a proposal. So after the breaking, and then at the points where there are breaks, new segments are going to be added. Okay, that's the dispensive model. The dispersive model. And then the other model that was proposed is known as semi-conservative replication. According to the semi-conservative replication, what hap- what they proposed was that when DNA replication is happening, these two strands that you can see will first separate. You can see the separation happening. This is one strand. This is again another different strand. And then they said that each one of these strand will now make a copy. Okay, will get a different strand. So you see this. And then this one also will gain another strand. So of these three models which were proposed, only one was correct. And the correct one was this one, which is a semi-conservative replication. Okay. Are we okay? Have I left out anyone? Okay. Please don't forget, semi-conservative is the method that DNA uses. Is there any question? Okay. Are you able to see my screen? Okay. Catherine, I saw your hand. Yes, Joe. Is it Joe? Joseph? Okay. Now, there's an experiment which was proved to prove. Okay. There was an experiment which was performed to prove that really DNA replication goes by semi-conservative replication. And it was done by two scientists, Meselson and Stahl. So we call it as the Meselson Stahl experiment. Meselson and Stahl experiment. You need to understand this experiment. It's very easy, but then you find those questions there in your exam. So now, so Meselson and Stahl, these two guys, in the year 1958, take note of the year. Okay, in the year 1958, they did an experiment and they proved that indeed DNA replication is semi-conservative. Again, what was the experiment for Meselson and Stahl all about? Proving that replication happens by what? Semi-conservation. Now, semi-conservation, semi means half. It just means that half of the DNA is coming from the original parents, and then half of the DNA is being made. Now, this experiment was performed using bacteria. The bacteria is called E. coli, E. coli bacteria. So, how was this experiment? What Nesselson and Stahl did is that they got E. coli and then they started growing this bacteria. So they got E. coli, they put it in an environment. Okay, this is an environment that has got nitrogen 15. Now, nitrogen 15 is heavy nitrogen. Please take note. This is what? Heavy nitrogen. So when they got bacteria and then they grew it in this environment, after a lot of generations, the bacteria incorporated the DNA. I mean, the bacteria incorporated the nitrogen 15 into the DNA, meaning that the DNA of the bacteria actually had nitrogen 15 in it. So, what Nesselson and Stahl did is that after they made sure that in all the DNA of the bacteria was labeled with nitrogen 15. They now removed the bacteria from that environment. They now took the bacteria in an environment that has got nitrogen 14. And this is light nitrogen. It's not heavy, it's light nitrogen. And then what they did is now they allowed the bacteria to start dividing. Okay, so they allowed the bacteria to start dividing, and the question they wanted to answer was, how is the DNA distributed? How is the nitrogen 15 distributed in the DNA of these bacteria? So now let's go on with the environment with the experiment. So what happened is that what they came to see, okay, the first parent, remember the first parent, bef- the DNA was labeled with what? Nitrogen 15. Please don't forget. Okay, let me put, I'll use blue for nitrogen 15 so that you don't forget. So the first parent had DNA with nitrogen 15. And then remember, the parent was removed from the environment of nitrogen 15 and was taken in an environment with nitrogen 14. So when this parent was allowed to divide for the DNA to replicate, what they came out to find, remember blue is meaning nitrogen 15. So what they came out to find was that half of the DNA, one strand, like here, had nitrogen 15, while the other strand had nitrogen. Actually, both strands had half nitrogen 15 and nitrogen 14. So you can see that each strand is a mixture of nitrogen 14 and nitrogen 15. Nitrogen 14 and nitrogen 15. Okay. And then, so this is after what? This is after the first replication. So from the first replication, what is the percentage of the new DNA that has been formed? You can see the new DNA is the one having 14. So I've got two 14s and then two 15s. So the total is 4. So if you get a 14, that is 2 out of 4, it is 50% new. Okay, 50% new strand. And then what they did is that they carried out another replication. That was the second replication, third replication, continued. So for the second replication, what happens that this guy separates. Okay, it separates. So it will separate into a brown strand. Even this one, the other one will also separate into a brown strand. But remember, they don't only have browny strands, they also have blue strands. So they also separate, also the blue strand is there, the blue strand is also here. So I've separated this one into brown and blue. Remember, brown is nitrogen 14, blue is nitrogen 15. I've also separated the other one. But remember, this bacteria is still in an environment of nitrogen 14 when this replication is happening. So what happened is that the DNA which was formed, the second replication, the DNA that is being formed is going to have a strand of nitrogen 14 because it's happening in an environment of nitrogen 14. That's why I'm using this color brown. Okay, you can see the new strand that is coming brown. Brown again, brown. Okay, we have formed a new strand. So you can see for these strands, there are four strands that we have formed in the second replication. But of the four strands that we have formed, can you see that two are completely new? They don't have any blue strand in them. So we have got 50% which are what? New. Can you see that two of them have got a mixture? So we have got 50% hybrid. Hybrid means a mixture. We have got 50% hybrid and 50% new. But then what is just the total percentage of the new DNA that we can see? So of this strand, we have got one which is new. Of this one, we have got one which is new. And then these, both of them, the two of them are new, the two of them are new. So that makes six of them being new. So after the second replication, we find out that 6 out of 8. So we say 6 out of 8 times 100. I think that should give us 75%. So we find out that 75% of the DNA is actually completely new, while 25% is old. So after another replication, the percentage of the old DNA will continue reducing and reducing. So the way they can ask you a question on the what I've explained here is they can ask you, according to Meselson and Stahl, after the first replication or after the first generation, the percentage of the DNA of the new strand which was seen is, you need to state the percentage. So I've stated for the first generation is 50% new. Second generation is 75% new, 25% old. Or if they are talking about completely new, like the completely new strand that is not hybrid. You have got these two, they are completely new, they are not hybrids. It's not a mixture of blue and brown. Is this clear? Is this clear? Okay, good. Now, what they did is that after they performed this, they actually used the centrifugation. All right, they used centrifugation. So what they did is that they got actually the bacteria, the bacteria which is here. They put in a test tube. After putting in this test tube, what they did is that now they spinned very much. They had to spin it very much. Now you know that nitrogen 15 is heavy, nitrogen 14 is light. So what happened is that on the uh, at the end when they looked at the test tube, they saw that for this one which was not yet taken into the first one which is not which has only nitrogen 15 before any replication had only one line. Okay, let me put the line a bit down. So I had only one line here. Okay, it had only one line, and this line is representing only nitrogen 15. But what they saw is that after the first replication, and after first replication, first, when they got to look at the test tube again, they actually saw that you have got different strands. You can see that both of them are nitrogen 15, isn't it? So the more you are on the left side, on the right side, the heavier you are, because this is the bottom of the test tube. So what they saw is that instead of the DNA being in line again, the DNA became a bit lighter, so it became a bit on top of the test tube. So they knew that okay, this one had a mix of nitrogen 14 and nitrogen 15, because the nitrogen 14 has made the DNA to be much lighter. That is why it is a bit on top. That's why it is a bit on top of the test tube. And then after the second replication again, they looked at the test tube. What they saw was they could tell that uh, one strand was here. And then they also saw another strand which was here, which was much lighter. So this strand which is here is a bit heavier. This is a mixture of nitrogen 14 and nitrogen 15. And then this one is only nitrogen 14 alone, which is much lighter. And these are the two that you can see. You have got this one and this one, which are mixtures of nitrogen 14 and 15. This is the one that you can see, that band. And then there is now these two, which are just totally nitrogen 14. These are the lighter ones that you can see here. So this is the experiment that Meselson and Stahl performed. Is this okay? Let me now talk about the last thing and then we are done. And this is the most interesting part of this topic, DNA replication. So, how does DNA replication happen? So, we are talking about the steps. So, all the steps that we are talking about in molecular biology, they are going to involve the first thing is initiation. Initiation is basically starting, all right? Starting. After starting, and then there's going to be elongation. You are now forming a new chain. After forming a new chain, and then there's going to be termination. You stop the new chain because you've already formed it. So these are the steps that to happen for DNA replication, transcription, and also translation. The same steps. Don't forget them, please. Don't forget them. Okay. So how does it start? So it all starts with DNA. This is our DNA. The first enzyme that is going to come into play is known as DNAA. So you see this enzyme like this, DNAA. What DNAA does is that it's going to come and locate the position of replication. So it locates the origin of replication. What we call as the origin of replication is a point where division is going to be happening. So DNA is going to be able to do that, to locate the point where replication is going to be starting, all right? And then the next enzyme is going to come into play. Then you are going to have another enzyme which is known as the DNA helicase. Okay, hope you are getting this. Okay, so DNA helicase is the next enzyme that is going to come in. But then what does DNA helicase do? Helicase is the enzyme that I showed you earlier that is going to come here. Helicase comes here and it starts to separate the chain. So what are you going to see when helicase starts doing its function? What you are going to see is this. Remember, one strand is supposed to be running in the 3' to 5', while the other strand is supposed to be running from 5' to 3'. And I'll be putting in red, that will be helicase. I don't want to be writing the name again and again. So what is the function of the enzyme helicase? Helicase is basically, remember you have got nucleotides. Like you have got a thymine. You know that thymine pairs up with the adenine. We did talk about this in DNA. And then you also have cytosine. Cytosine pairs up with the guanine. If we add again a guanine on top here, guanine pairs up with cytosine. And then if we add again adenine there, adenine pairs up with thymine. So helicase is basically breaking phosphodiester bonds, because the bonds that you find between nucleotides is a phosphodiester bonds. Phosphodiester. So what's a function of helicase? Is to separate the DNA strand. How? By breaking the phosphodiester bonds. But then there's something that is a bit interesting here. Okay? While this is happening, you know, while helicase is separating, the way they say it, deshtanga nabomba fyabomboloka. Hope you can get that. So helicase is separating, and then the DNA can be again connecting at the back. So to prevent the connection at the back, you are going to have some other enzymes here attaching. So these enzymes, we call them as single stranded binding protein. So we'll say SSB. So you normally see SSB protein. So that is a function of single stranded binding protein, to prevent the DNA that has been separated from coming to connect back together. Now, can you see that DNA has got supercoils? Look at the front here. It has got supercoils. So you want to remove those supercoils so that it can be easy to separate the DNA. Another enzyme is going to come in. What is the enzyme really doing? It is removing supercoils. So how you want to make the molecule to be is like this. Can you now see how it is? Instead of the molecule being coiled, it is now a bit straight. So it will be easy for helicase just to come and separate. So if this is the 3' end, this is going to be the 5' end, 3' end, this is the 5' end. Remember, the guy helicase is still there. Here is the guy helicase. Okay. So the enzyme that was removing the supercoils, we call it topoisomerase. Topoisomerase. It has got another name that you'll see in the exam. It's also known as the DNA gyrase. So what DNA gyrase or topoisomerase does is that it comes and removes supercoils so that it can be easy for helicase to work. Now let me ask you, Are we okay up to this end? Have I left out anyone? Yes, Joseph. We are good. Be free to ask. Okay, be very, very free to ask. Okay, good. Let me now jump to the next page because there is no space on this page. Don't forget so far the function of helicase, the function of DNAA, the function of SSB, and the function of topoisomerase. Now, what is going to be happening is that after you separated your molecule, another enzyme is going to be able to come in. So, how is our molecule now at this point? Our molecule is now going to be like this. So, this is our DNA molecule at this point now, which has separated already. Let me make it nice so that you should be able to remember. Okay. So these two lines I'm representing, they are writing, they're representing nucleotides, the adenine, thymine, guanine, and the cytosine. Remember, we don't have uracil in DNA. So one strand is running from 3 prime to 5 prime. Another strand is running from 5 prime to 3 prime. So when helicase is separating this strand, another enzyme is going to join in, and that enzyme is going to be following at the back. So helicase is going forward like this, right? Another guy comes in, and the other guy is known as DNA polymerase, and specifically it's DNA polymerase 2. So what DNA polymerase 2 does, now before DNA polymerase 2 comes in, you are going to have what we call the RNA primase. Okay, please take note. RNA primase. What is the function of the enzyme primase? Just from the word primase, it is there to just add a primer. Okay, a primer. A primer is just a nucleotide. Now, primase, what it is going to do is that it will just add one nucleotide on the strand that is running from 3 prime to 5 prime. Please take note of that. So on this strand, which is running from 3 prime to 5 prime, RNA primase is going to lay one, please switch off that mic, one primer. So one primer for which strand? The strand that is running from 3 prime to 5 prime direction. But the same RNA primase is going to lay down a lot of primers. A lot of primers. Joseph, switch off your mic. For which strand? For the strand that is running from the 5 prime to 3 prime direction. I need you to take note of this, very important. So for this strand that is running in the 5 prime to 3 prime, primase is going to put a primer here. It will put a primer here. You can see the primers are a lot. But for the previous one, only one primer has been put. And then now another enzyme is going to come in, and that's the enzyme that we are calling as the DNA polymerase 2. Okay. So DNA polymerase 2 is a good guy. DNA polymerase 2. What this guy is going to do is now to continue to form phosphodiester bonds. It will continue to form a new strand. So for the strand that is running from 3 prime to 5 prime, you can see the way it is doing. So this is the guy. It is going in the direction of the replication fork. Remember, the replication fork is going forward. Since this chain was in the 3 prime to 5 prime, DNA polymerase, the chain that is forming is going to be from the 5 prime to 3 prime direction. So you always form a chain in the opposite direction. So if let's say the chain that we started with was running 3 prime A, G, C, C, T, G, A, C, G, T, 5 prime. So DNA polymerase, the new chain that is going to make 3 prime pairs up with 5 prime. A always pairs up with T. G pairs up with C. This C will pair up with G. G pairs up with this is another C to pair up with G. T pairs up with A. G pairs up with C. A with T. C with G. G with C. T with A. And then at the end you put 3 prime. So just know that A always pairs up. It uses 2 double bonds. A will hydrogen bond, sorry. So A will pair up with thymine. And then cytosine pairs up using 3 hydrogen bonds with guanine. So this is what happens. Okay, that will be the strand that will be forming. Now you need to know that DNA polymerase 2 works in both directions. It will be working in a forward direction at the same time it is working in the backward direction. But remember for the backward direction, so the strand that is running in a forward direction, the strand that moves from 3 prime to 5 prime, we call this as the leading strand. And the leading strand is made in a continuous manner. DNA polymerase will continue making phosphodiester bonds continuously. But for the DNA strand that is running from 5 prime to 3 prime direction, this strand is known as a lagging strand. It remains behind. Why is it known like that? That is because what the guide DNA polymerase does is that it will be making only lead to few uh, bonds here. Okay, you can see it is just making, it is not fully making bonds. Remember, they already primase added a lot of primers for this strand, which is the lagging strand. And then DNA polymerase doesn't come and form phosphodiester bonds for the lagging strand. The function of DNA polymerase for this strand is to form what we call Okazaki fragments. Okazaki fragments. So Okazaki fragments are just small, like you are just forming small uh, nucleotides. Like you just add a bit, you add this one with this one, and then you skip a bit. Okay, you add these two, you skip a bit. These are Okazaki fragments. They are fragments, remember. So DNA polymerase 2 for the lagging strand forms Okazaki fragment. The enzyme that will form phosphodiester bonds on the lagging strand is known as the DNA. The previous one I talked about was what? DNA gyrase. Okay, this is now known as DNA ligase. DNA ligase. Please don't confuse it with DNA gyrase. So DNA ligase is forming phosphodiester bonds on the lagging strand after DNA polymerase raised down Okazaki fragments. So in other words, those to my few things that DNA polymerase was doing, it puts this one, it puts this one, it leaves a space, it leaves the space. Then I guess is going to come and now make phosphodiester bonds, forming full bonds. But take note that for the leading strand is DNA polymerase, which is forming the phosphodiester bonds. DNA ligase forms phosphodiester bonds on the lagging strand. It's different from DNA gyrase, which is also known as topoisomerase, which was removing the supercoils. So this is the process of DNA replication, and we are done. Just know that for the DNA polymerase, we have got different types. We have got DNA polymerase 1, DNA polymerase 2. So DNA polymerase 1, polymerase 2, and polymerase 3. So DNA polymerase 1, this one has got what we call exonucleic activity. Exonuclease activity. The meaning of that is that it basically corrects mistakes. Okay, so it corrects mistakes. Same with this one, it also has exonuclease activity. except this one only correct mistakes in the 3 prime to 5 prime direction. Let's say, for example, when you're adding the nucleotide, instead of adding adenine, you added the thymine. So what happens that DNA polymerase 1 and DNA polymerase 3 can come and correct that mistake. But the DNA polymerase 2, remember this one was involved in what? Polymerization. To form a polymer, to form that long chain by adding the forming the phosphodiester bonds. So this is DNA replication. The next topic we are talking about is DNA transcription, and then DNA translation. Then we are done with molecular genetics. Do you have any questions before we cancel the class? So if you're not in the group, make sure you inbox me. I add you in the group because that's where I'll send the questions. Is there any question? We are good? All right. So thank you so much. Enjoy your night.